Hello, everyone, and welcome to this new edition of the Impactive webinar series. As it's been the case in the last few webinars, we're here to introduce our sister projects. These are different projects funded by the European Commission, which pursue the same goal, pursue the goal of making greener pharmaceuticals. Today, it's a pleasure for us to have the project ENVIROMED, which is a Horizon Europe initiative pioneering AI-driven process control, sustainable manufacturing, and innovative monitoring of pharmaceuticals and manufacturing tools to reduce the environmental impact of making medicines throughout the whole life cycle, basically. So, like you very well know, if you're watching this webinar, Impactive is also a Horizon Europe project. You can follow us on mechanochemistry.eu, and you can follow us on our social media. And today we'll have a little time for questions and answers at the end of the webinar after the presentations. For that purpose, if you could please put the questions and write the questions down on the chat available for that purpose on YouTube, that'd be fantastic. And without further ado, I'm just going to quickly introduce our two speakers today. It's quite a special day because we've got two people presenting, both from the Horizon Europe sister project ENVIROMED. So first off, we're going to have Stephanos Camarinopoulos, who's the environment coordinator and also the director of RISA. And basically in RISA in Germany, they carry out a range of activities from power plant safety assessments to advanced environmental information system. Stephanos has over 20 years of experience in the delivery of software solutions, driving cutting-edge research and commercial software solutions to the market. They have a very clear strategic vision, uniting technology and innovation to create sustainable and at the same time very impactful results in pharmaceutical manufacturing. And then also from ENVIROMED, we're going to have Michael Harasek, who's a full professor at the Institute of Chemical, Environmental, and Bioscience Engineering in the Technical University of Vienna in Austria. There in Austria, he leads research unit in thermal process engineering and simulation, which is focused on membrane separation, computational fluid dynamics, and sustainable process design. Like you see, sustainability is the key issue for assisted projects and also for Impactive. Harasek has also received several awards, including the Hauska Prize and the Best Patent of the Year in Austria. So we are very, very happy to have both of them here today representing our sister project ENVIROMED. It's great for Impactive to have these collaborations with the projects pursuing the same sustainable goals in Europe. And yeah, I think I'm just gonna go forward and continue with the presentations. We're gonna hear from Stephanos first and then Michael. So then please, Stephanos, the floor is yours. Everyone in the audience will keep the questions and answers to the end of both presentations. If you've got any questions during Stephanos' talk, please put them in the chat and we'll address them at the end of the talks. Sorry about that quick notice, but then Stephanos, now the floor is indeed yours. Sorry.
Thank you very much. Thank you very much for the introduction, and good morning also from my side. We are very glad to given this opportunity to being here today, and we really like the initiative that the Impactive project took and organized this series of webinars. We really enjoyed also viewing the previous one that you organized, and be happy also to share our project and the results in this framework. So let me quickly start with a quick overview of environment. The duration of the project is 42 months. It started back in June 2022, and it is planned to be completed this year in November. The overall budget of the project is 7.5 million euros, and the consortium consists of 18 partners, out of which 8 are SMEs, 5 large enterprise organizations, 5 university and research and technology organizations, and our partners are spread across Europe in 11 countries. Having said that, let me give you an overview of the main objectives of the environment project, which includes, First of all, to enable a better understanding of the environmental impact of pharmaceutical compounds throughout their life cycle, from the design and the fabrication of the and the use of this and disposal. Another main objective is to support the reduction of the environmental footprint of the manufacturing processes, and of course, finally, to contribute to the scientific knowledge to support adaptation of existing regulations and processes. So, in order to achieve our objectives, there are a number of methods and technologies that have been developed in the context of the project. As you will notice, while I will give you a brief overview of them, Environment is a multidisciplinary project covering many different research and application areas. So as you can see in the figure, our project activities span across all stages of the pharmaceutical life cycle, namely the pharmaceutical development and manufacturing, the use and disposal of pharmaceuticals, and the ecotoxicity assessment. Now, let me briefly present the technologies developed in the environment. First of all, we have developed in silicon tools for green drug development. These in silico tools evaluate the compliance of chemical compounds with green chemistry principles for green by design pharmaceuticals. The machine learning models that enable the prediction of potential ecotoxicol- bah, ecotoxicological outcomes have been successfully developed and, of course, also validated during the project. Moving on to our next technology, we have developed a digital transformation toolset towards a robust and efficient continuous biomanufacturing line that overcomes the problem of batch production using advanced sensing, AI and machine learning algorithms, and digital twin models for better prediction and control. We have also developed spectroscopic techniques for manufacturing and processing monitoring. Specifically, these techniques enable the continuous monitoring of all the manufacturing processes, and the reduction of the use of chemicals. The first prototype of the so-called liquid monitoring analyzer has been released and is currently being validated. Another important aspect of our project is using mid-infrared polymetry for vessel inspections, aiming to result in the reduction of the rising cycles and cleaning solvent usage. To this end, we have developed the surface inspection analyzer, which is also currently being validated in collaboration also with our pharmaceutical partners, Pfizer and Novo. Moving on, another application of the laser-based spectroscopic in our project is an online monitoring of wastewater. Specifically, a wastewater spectroscopical analyzer has been developed to enable monitoring of effluent of clinical facilities and wastewater treatment plants input-output to accurately map concentration levels of pharmaceutical micropollutants. Another activity in the project, which is also related to the wastewater analyzer, is the organization of monitoring campaigns that enable better understanding of the severity of the contamination at the various stages of the pharmaceutical life cycle, production, use, and disposal. To be more specific, we have organized 3 environmental monitoring campaigns that took place in Greece and Italy. They are conducted by the Athens Water Utility Company EIDAP and Mitera Hospital in Greece and the CNR in Italy. The process involved sampling at both the entrance and the exit points of EIDAP wastewater treatment plant, and then the influent of Mitera, and conducted a detailed chemical analysis to accurately assess the presence of pharmaceutical micropollutants. In parallel with these campaigns, a marine monitoring campaign is also conducted, aiming to have a better understanding of the pharmaceutical effects in the marine aquatic environment. To this end, a DAP collected near to the wastewater treatment plant not only water samples but also marine organisms and ships them to the CNR in vivo to conduct in vivo ecotoxicity studies. Uh, it's also worth to mention to say that besides the in vivo ecotoxicity studies, we performed also in vitro ecotoxicity studies. This aim to assess the effect of long-term pharmaceutical accumulation. Yes, also in the context of the project, we have developed a registry platform of pharmaceuticals, micropollutants, which is actually an enhanced version of the pre-existing registry that we had, as it has been enriched with additional parameters and functionalities based on the project's findings and pharmaceutical partners' feedback during the project. List and but not least is our project performed also a LCA analysis focusing mainly on the manufacturing stage of the pharmaceutical production products and proposing greener alternatives compared to the conventional industrial processes that are used today. So now let me give a brief overview of the three novel monitoring instruments that we have developed within the project. First of all, the wastewater spectroscopic analyzer that enables near real-time monitoring of the pharmaceutical micropollutants in the wastewater. The analyzer is currently used in our monitoring campaigns. The second instrument that is developed in the project is the online inline monitoring analyzer, which obtains real-time information on the chemical composition of the liquids that are found in the pharmaceutical reactors during cleaning reinigung steps. It enables process control optimization, for example, the reduction of reasoning chemicals and cycles that are needed. Finally, the third instrument is a surface inspection analyzer. This analyzer is applied to probe surfaces of pharmaceutical production vessels to check for remaining substances. It facilitates a decision on whether additional reasoning cycles are necessary or not. With this, it enables a more efficient reasoning and reducing the use of chemicals in the production phase. The three analyzers that we have developed use specially designed lasers that were also developed within our project. Moreover, it is also worth mentioning that at the end of the project, we aim to have reached for all three analyzers TRL level of 6 and are optimistic, and we are already we have plans that beyond the project end, we are going to make these instruments ready to reach the market, as we have already get some very positive feedback about our technologies by our pilot partners. Speaking of our pilot partners, in this slide you can see a summary of the pilot activities that are currently running in our project. So we have our two pharmaceutical partners, Pfizer and Novo. They are involved in the testing of the liquid monitoring analyzer and the surface inspection analyzer against the current cleaning verification methods that they are applying today. Our other two pilots are Mitera Hospital and EIDAP wastewater plant in Psytalia, where at Mitera Hospital we are installing the wastewater analyzer to track wastewater streams and detect pharmaceutical micropollutants. And the EIDAP pilot involves the installation of the wastewater analyzer to monitor influent and effluent in terms of concentration of the targeted pharmaceutical compounds. So that is from my side, and I'm happy to answer to any questions that may rise after also Michael's presentation. So Michael, I think the floor is yours.
Thank you, Stephanos. I think we just need a quick second to switch the slides, but then of course,
Okay.
like you said, Michael, the floor is yours. We really look forward to your presentation, and we'll hold the questions until the end. For anyone who's joined halfway, I will be taking questions from the chat here on YouTube. So please feel free to send those, write those. We'll be monitoring them for the Q&A later. And yeah, now, Michael, indeed, the floor is yours. Looking forward to your presentation.
Mm-hmm. Yeah, thank you. Thank you very much to the IMPACTIVE team, project team, for this great opportunity to present some of our results. I wish all the audience a wonderful afternoon. And in the upcoming minutes, I will briefly present some other aspects. Stephanos has already mentioned one of the core activities is actually sensor development, but we have also, together with the team here at TU Wien, also looking at bioprocessing, bioprocess design. And I have called my talk today Bioprocess Design and Control from Digital Twins to CFD as Computational Fluid Dynamics as Process Lens. The project has already been introduced very well by Stephanos. So the focus of today's talk is actually to look at the impact of the processing, not so much the impact on the environment, but rather in its use along the whole. I have split my slides into two topics, and the first one is the bioprocess control development. And here I have to I'm very happy to present results from Rüdiger Lück, Julian Kopp, and Oliver Spadiot, who are all here at TU Wien with in their own research unit bioprocess technology. During this project, we have actually decided to run a lab scale or bench scale fermentations in order to apply and develop our technique. And here the idea was to actually move from a fat batch fermentation, which has, of course, longer downtimes, so a continuous operation for the project and for the model developments. So moving from a classical process as it's used nowadays in many bioprocessing and biomanufacturing facilities towards a continuous operation was one of the targets. The microbial host that was selected for this purpose is based on E. coli, Escherichia coli, and there is a special recombinant fragment antigen binding FAB molecule that is the target molecule or the API that was selected during this manufacturing process and during these studies. And it is based on previous work here at TU Wien published by Gundinger et al. And I have also provided to you the link and some background information on that. Yeah, when we talk about sustainable bioprocessing, it's always a question, what does it mean? What does greener biomanufacturing mean at all? And it is about environmental impact reduction of the bioprocess, measured with traceable indicators. And these indicators are based on the input and output in relation to the material flow, the mass flow, the energy flow, the waste flow, the CO2 emissions, and in relation to the target variable, which is mostly in biomanufacturing the final product titer. So that means the concentration of the product that we can achieve with all the efforts that we take in such a production line. So as such, we can actually also apply green metrics, the energetic footprint, the space-time yield, a very important parameter in particular with the expensive use of fermenters in particular, the process mass intensity, and of course also the CO2 footprint. Now, giving you a quick insight into what we did here during the last years with regard to the fermentation. Here, what you can see here is are some results. And of course, when we talk about control of fermentation, it's always a question which parameters to look at. Is it the CO2 emission? Is it the glucose concentration? Or in our case also, it can be actually the phosphate concentration, which is consumed by the microorganisms. For a classical fat batch fermentation, for those of you not so familiar with it, we usually it is started with a batch phase to grow the microorganisms, then having a fat batch phase, and then continue with actually for a while with a fat batch phase. And here it is important to find the right timing for the harvesting of the product. We want to have the maximum concentration and no drop in that concentration. So in order to develop control options here, it is important to actually run a variety of different operations. So to have a design of experiment where the temperature, the feeding rate, media composition, the harvesting time point is varied. And that is actually fed into a machine learning trained digital twin to have a prediction of the dynamic process based on certain parameters that can be easily measured. And of course, also based on the experimental data, the continuous forward training, also with new results, is one of those aspects that are developed in the team of Oliver here. So looking at the fermentation, at this fat batch fermentation, you can see here some parameters, the concentrations as they develop. The biomass is of course important because here that expresses also our product, the glucose concentration, phosphate concentration, which is quite low and is actually limiting the growth rate. And the CO2 off-gas concentration and its development is also an indicator of metabolic activity and biomass growth. So from the model, actually, we can, and with this trained model, after the DOE training, we could show that we can predict the batch end very precisely, also biomass growth inhibition if it happens at all, the glucose accumulation, but also product formation kinetics. And if you look at that into that here, that is the concentration of the biomass here that is given. And the question that is of course important to optimize and also minimize the resource consumption is to have the optimal harvesting time point, and that could actually be achieved in various runs, which we could demonstrate where we applied our digital twin control here. Moving to continuous processing, that's the other aspect here of this work. And this graph just shows you how a typical continuous operation with the knowledge of the fat batch could actually be imposed. So starting with the batch of fat batch and then having a continuous harvesting of the product and the biomass. And This just shows that it can be established, where we control the dilution rate and the phosphate concentration as a limiting parameter here. Moving now to and quickly introduce you to also our CFD activities that accompanied the work that was done in the lab of Oliver Spadiot here at TU Wien. We look into during this project we looked into CFD modeling. Those of you that are not familiar so much with CFDs, computational fluid dynamics, is a method that allows to calculate the flow fields in complex environments. And if you consider a fermenter that has gas sparging, that has various levels of stirrers, that has also a certain liquid level, that has changing viscosities, that has biomass expression, then this is quite a challenging task to have a proper representation of such phenomena. So all in all, within ENVIROMED, the computational fluid dynamic simulations that we did were also validated by the experiments that I briefly introduced you to. And the targets that we had with the CFD simulations were to analyze the fluid flow, to learn more about the velocity fields, to get information about shear stress, which is highest at the tip points of the stirrers usually, but also about mixing times. And the mixing times are important also to have a proper sensor placement, for instance, in order to add the carbon source or also trace components in a proper way and have the sensor in the right spot. This workflow that we set up was based on an automated geometric importing, and then these procedures in order to discretize the governing equations in computational fluid dynamics usually require a meshing, which is also automated, and we have also automated post-processing developed in this context. Our approach in ENVIROMED was to use open source tools that can be also disseminated and are easy to access, so there is no commercial CFD software used. So we decided to use OpenFOAM, which is a well-recognized open source CFD code, C++ CFD code, and so we developed the workflow for our needs here that includes the geometry implementation, the pre-processing, it's the meshing, a decomposition of the geometry for parallel processing on high speed on our high-speed clusters, as well as the proper selection running of the solver and the post-processing to get results like the velocity and turbulence parameters, the contour plots, ISO surfaces of volumes, line plots, torque, energy consumption, or mixing quality. I just don't want to tell too much and talk too much about the details, but we have also reported a decision tree for the proper solver selection, depending on the task, on the simulation task that we have in the specific project. Just to show you some of the results, it's just some flow feeds that you can see here. These 3D flow regimes, of course, also can be represented in videos. We can analyze where we have very low velocity, so these are dead zones, or also the level of the shear stress. With a transient experiment, we can calculate mixing times and also consider non-Newtonian rheologies along a fat batch fermentation, for instance, where we have low biomass concentration at the beginning, but that increases during the fermentation. So here, different states of that can be actually represented in the simulation as well. The prediction, for instance, of the power consumption of this multi-force fermentation system that I briefly showed to you, that's here to be seen on the left side on this slide here, is quite well. So actually we can calculate the mechanical forces, and we did also measurements on the electricity and on the torque of the actual system. And with this, we can actually also predict scaled fermentations with regard to its energy consumption. Let me briefly summarize and conclude. We have shown, or I have shown here shortly in this presentation, that we have developed a bioprocess control system that allows to predict the performance and analyze the performance of fed batch and continuous fermentation. And for the CFD work, we have implemented the workflow for steered bioreactor systems that are based on open source tools, OpenFOAM, and also some CAD tools. And we have also validated the results with power consumption trace experiments. And at this end of this short presentation, I'm happy to pinpoint you to our recent review paper that was just published a few weeks ago, which is called "The Potential of CFD in Sustainable Microbial Fermenter Design." Would be happy if you read it, and also get in touch with us in case there are questions. With this, I'd like to thank you for your attention. I'm ready for any questions.
Well, thank you so much for the very nice presentation. And I think now we're gonna have questions for both of you. So feel free to if one answer each, or just answer depending on your expertise and your involvement in the project. I'm sort of have a couple of more generic questions that could be addressed to Stephanos as coordinator of ENVIROMED. You mentioned already doing some tests with real pharmaceutical companies. I saw Novo Nordisk and a couple others in your slides. What can you say about having technology ready for the market at the end of the project? How are these collaborations with big pharmaceutical companies going?
Okay, that's a tough one. Indeed, we test our systems at the pharmaceutical companies. However, as it is a research project and we are developing the analyzers and the lasers within the duration of the project, and it's foreseen that at the end we are going to reach TRL 6. What we have accomplished is that we have proven that the technologies are working. The results are almost what was expected to be. And we have acknowledgement from the other partners that we are on track, and that's what has to be done. So there are already some plans in place to further develop, further test, and potentially bring these technologies to the market with a short period, 2-3 years after the end of the project. Of course, there are some limitations, some constraints. Funding has to be there, but we are optimistic that we will overcome these challenges and that the partners will accomplish their objective to bring them to the market.
I think that's very, very interesting. And then also, just for people around the audience who are not familiar with the concepts of TRL, I think TRL6 means that basically the system works in a relevant environment. So in your case, you're already demonstrating that with big pharmaceutical companies. So I would say that you're meeting the challenge, and you have something that is very, very interesting there. You mentioned there's a couple of challenges to move this to the market and to real life. You mentioned there's a little bit of a financial constraint, but then also what about the regulatory constraints? Because in our case, in IMPACTIVE with mechanochemistry, changing the whole processes by which we manufacture pharmaceuticals is always a challenge. I don't know if you've noticed something similar in ENVIROMED. Do you see any backlash from the pharmaceutical industries to modify the process and update their equipment?
I think maybe Michael, you are.
Yeah,
You
Michael,
bring
feel free to
Yeah,
feel
yeah,
free to
yeah.
answer.
you
No,
bring the solution there.
I'll bring the solution. Oh well, I think what has to be understood is the pharmaceutical industries, and that is also our experience, is of course very conservative, but conservative in a sense that based on the regulatory decisions, there is an established process, and so to bring in greener manufacturing methods, whether it is an improved process controls, whether it is a new sensor, or whether it is a new algorithm that improves, or also a change from a fat batch, for instance, to a continuous process, is a very strategic decisions by the pharmaceutical companies. And I think what is very important actually is to keep the discussion and the information flow. And at some point, I believe when a certain technology readiness is reached, for the sensors, for instance, an acceptance by the industry would definitely happen. But it requires resilience on all sides, in particular also on the universities, because our developments that we have usually end at a low TRL, TRL 3, 4, 5, maybe at most. So here that gap between an acceptance in the industry is for sure a long journey. But I believe it's positive to work on these topics and also to keep the discussion and the information flow to the industry.
I think that's a very good point. I had another question for you, Michael, regarding more of a technical side. Because obviously, I think this may be one of the good uses of AI and computing, trying to simulate things that otherwise would be very expensive to simulate in the lab. Could you comment a little bit on your progress, not only on CFDs, but I was very interested on the presentation slide when you mentioned like digital twins, because that's a very interesting tool that's being used across disciplines, not only in chemistry and manufacturing. Could you give a little bit of an overview of that and what the challenges have been so far to develop digital twins of engineering processes?
Yeah. Well, what I showed today is actually the process control of a bioprocess, and the challenge here is actually access to the data. So a well-trained model, whatever algorithm is used in AI, requires a strong data set. So that means also a variation of parameters, and that is sometimes tricky, in particular for biomanufacturing. For a classical chemical manufacturing, it's easy. We can change the temperatures or something like that, and change rates, and then something happens, and we get a quick response. In biomanufacturing, considering that the growth of the microorganisms takes time, and also the expression of the product takes time, and also reaching conditions that are harsh or not preferential to the microorganisms takes much more work. And so also here it's I would say a lengthy journey to have a proper and well-trained model available. And my colleagues here in bioprocess engineering, they have a long track record and experience, and also developed some software tools that support the way. So a proper design of experiments for training such a model is essential. Further, it's also important to have a representative sensor implementation. So you can only control where you have information about the process. So that means what do you sense? Is it the glucose concentrations? Is it the biomass concentrations? Is it the turbidity? Is it the pH? Is it a trace component that can be measured? Or a multitude of everything? Or is it infrared spectra? Or is it other information that all can go into such a model? And here it is actually a compromise between the data that can actually be implemented in such a model and can be used online in such a model. And actually the outcome, which is the control of a dosing, of a stirrer speed, of the temperature, or a decision towards harvesting, or whatever is relevant in biomanufacturing. So this is, I believe, a long journey. In computational fluid dynamics, it's a little easier. Yeah, we can run also variations and see in which direction certain aspects go. Also geometry variations. So what is the best positioning of a stirrer? What is the best geometric design? How does that work? Or what happens if we increase the oxygen or air dosing or things like that? It's a little easier and here to harvest results, but it takes time because it is computational demanding, and such. It's not for real-time control, but rather for an in-depth analysis of equipment. That's my opinion.
Yeah, and in general, maybe going back to Stephanos, reducing the overall environmental impact of pharmaceuticals throughout the whole life cycle, it's a big challenge. So what do you think is more interesting in this challenge, and then also what aspects of the whole sort of life cycle are you focusing in more in ENVIROMED?
Okay, of course, our project involves all stages. So we look on all of the stages. Of course, they are all important. Given but the limitation that we are a research project, we have 3.5 years, it's very difficult to provide solutions for everything. So I think what we have accomplished, we are focusing more in the manufacturing stage, where we have identified some key factors where we can be more green. The energy is something very important. So we monitor the energy and see how we can reduce the energy consumption during the production phase. This is one of our strong outputs, I think, from this project.
Any comments, Michael? Like, what do you think are the more challenging parts of making pharmaceuticals more sustainable?
Two aspects. It depends. First of all, it depends very much on what kind of API are we considering. Whether it is a super low concentration API like that what I showed today, that is expressed by microorganisms in the microgram or milligram per liter concentration range. So here I believe that increasing the titer is one of the core targets, because that means that the downstream processing, which may take 50-80% of the efforts in the final product that goes on in the market as a medicine, that this is impacted very, very strongly. Like in contrast, as in your project, in the project IMPACTIVE here, mechanochemistry technologies means actually to be solvent free. So that means having the highest titer possible. Which is not possible with microorganisms. It can be increased, but not so much. So I think it depends very much with the biomanufacturing, and to my experience here, increasing the titer, reducing the fermentation times, so that having higher growth rates, having the best available conditions for microbial growth and API expression is for sure one of the core targets. And considering also novel methods, which was not analyzed here, which was not part of this project here, but to look at downstream processing in particular also. And here, this is another aspect. Moving from fed batch or from batch to continuous also reduces the impact very much, because the cleaning, the one of the most expensive and energy resource expensive products is actually water for injection. So that means in the cleaning cycle of fermenters, we have a good chance by moving from a if possible, from a batch to a continuous operation, actually the cleaning cycles are much, much less prominently implemented in such processes. So that is also one of the, I think, one of the advantages. But that's a paradigm change, yeah? And it's not possible with all the production lines, and it has also to go in hand in regulatory approaches.
I think that's a very interesting comment as well, because we always think of drugs as like just the pills that we take, but then obviously there's so much more, like you just said, the cleaning, the packaging, the excessive prescriptions, and then sometimes we end up with a bunch of drugs that we don't really need. So it's not only making them the more sustainable per se, but also making them more sustainable throughout the whole like life cycle. From being more cautious with prescription to maybe manufacturing methods that are a little bit more, on demand and more sustainable. I was gonna just quickly, just because I don't want to have this webinar run too long, just wondering if you have any closing remarks, any I don't know, couple of things that we may not have mentioned, and you think they're key for ENVIROMED and for this family of EU funded projects. Either of you, I'm happy with Michael.
Okay, one remark that I have, that I see a difference in compared to previous project that we have coordinate or participate, is that we have created this green pharma cluster in this with the sister project, and we have the opportunity to exchange ideas, share results, learn from what the sister projects have done, get their feedback, and be more united and go united to the commission and ask for help where we need. And we have also identified that we share the same difficulties, although we don't have exactly the same scope within the project, but we share the same difficulties. And all this discussion that we had during the last 2 years, also with the EU project officer and from the policy makers, it helped for us, at least it helps a lot, and I think we gained something more. Yeah.
I think that's a very interesting point. We also, benefit a lot from collaboration because, like you said, we share a common goal of sustainable drug manufacturing, and I think it's good to see this trend because in our experience as a company that does science communication, we've seen many projects and many other calls foster this type of collaborations across different projects. And I think it's a very good idea. Sorry, Michael, I think you wanted to add something.
No, just to add, I think to have this cluster and also to have an exchange with the sister projects is essential, because it strengthens all the individual projects, but also we have a common voice. And I think that is very important, both against or towards the industry, of course, that we want to have in our boat. We are sitting in the same boat, kind of, but the understanding is always a little bit difficult here. But also regulatory bodies and the commission itself. I believe that that this sector here, the biomedical sector, the medical sector, will definitely be one of our big cruise ships in Europe. Here we are strong, and I think it's important to strengthen the strength and continue in this regard, and also exchanging within the sister projects. It was very exciting to see. I'm a chemical engineer, so just as from the chemical engineering viewpoint, also. What are the other approaches? How do we meter? How do we benchmark our processes? What is green metrics? I think all this is very, very important and is essential for and beneficial for all the partners, I would say.
Yeah, I think you also mentioned before that it's important to keep like everybody informed, including like policymakers. So in this case, it's good that we talk between each other because we don't want to be speaking about TRLs or green metrics or things like that are not put in common before we actually speak about them, because otherwise you may think something is green, but then it's not because we're not following the same scales. So I think there are two very important points to close. I just want to thank you both again for joining us in this webinar session. And just, a quick reminder that people can find you online. I know you're on LinkedIn, you've got a webpage. So if you're interested in ENVIROMED, please look them up. They're very, very active, and they also have a fantastic communication team. And if you're interested in IMPACTIVE, which is the project organizing this series of webinars as part of the communication dissemination activities of this sort of family of sister projects, please follow us on mechanochemistry.eu. Subscribe to our newsletter to get news about the upcoming webinars. I think we've got one coming up at the end of November, so there's a little bit of a break there to relax. But we'll be back with like fully charged batteries and fully charged energy to talk about sustainable solutions for green pharmaceuticals. Again, thank you both for joining us today. It's been wonderful to see your presentations. Thank you everyone who joined. This will remain on YouTube for posterity, so people can go back and check and potentially contact us if they're interested in sustainable solutions for pharma. Thanks again, everybody, for joining, and yeah, see you soon.
Thank you.